Hello everyone, welcome to ITLS Academy. This is continuation part of early development, and in today's lecture we will see what is the cell cycle. Cell cycle. The very first point is the cell cycle includes four main phases. These four main phases are These are the four phases that are present in the cell cycle. If we talk about the G1 phase, then this is gap 1 phase, and this is the phase in which cell maintains its shape and size. Next one phase is S phase. In the S phase, cell synthesizes its DNA content. It replicates in DNA and synthesizes DNA. The next one phase is G2 phase. Again, in this phase, cell growth and cell shape and size is maintained. The M phase is the main phase in which division of any cell takes place. This M phase is further divided into four phases. First one is prophase. Next one is metaphase. Another is anaphase. And the last one is telophase. These four phases are present in the M phase and are responsible for the proper division of the any cell. Although this M phase lasts only for 5%, it only covers 5% portion of the cell cycle. The whole 95 portion completes this G1S and G2 phases. So let us see how these four phases are described in this slide. DNA synthesis, that is S phase. G2, it is a gap phase during which cell increases in size and prepare to divide. The cell division phase is also called mitosis or M phase. And another one is G1 or a gap phase, and this is also a phase for the cell growth and replication of the centrioles. This is the main point, that replication of centrioles occurs in the G1 phase. When a cell exits the cell cycle to differentiate, for example, it is said to be in G0 phase. If I draw a cell cycle here, this is our cell cycle, that is from G1 to S, from S to G2, from G2 to M. But whenever there is any cell that exits this cell cycle and it comes into G0 phase, it stops dividing. All these cells that are present in normal cell cycle, cell division pathway, those will divide. But those cells that skips this cell cycle and comes in G0 phase, these cells stop dividing. The division capability is almost lost in these G0 stage cells. Although we can recover the dividing capacity whenever there is any need in the body, in the case of like any replacement of cells is needed or any other thing, we can reactivate these G0 cells stages for the division. So in brief, we can say that a cell cycle is a series of events that takes place in a cell as it grows and divides. A cell spends most of its time in what is called interphase, and during this time it grows, replicates its chromosome and prepares for cell division. Next point is particular CDKs, that is cyclin dependent kinases, and their associate cyclins regulate the transition from one phase of the cell cycle to the next. CDKs or cyclin dependent kinases are the families of protein kinases, and these are first discovered for their role in regulating cell cycle. These CDKs are also involved in regulating transcription, mRNA processing, and the differentiation of nerve cells. By definition, a cyclin-dependent kinase binds a regulatory protein called cyclin. Here, a very beautiful diagram of cell cycle is shown. You can easily understand the steps or phases that are involved in cell cycle. This is our actual cell cycle image that will clear all your doubts regarding the cell cycle and all its phases, that what are the activities that are performed in every phase, what are the actual functions that are performed during the gap 1 phase, gap 2 phase, and in the synthesis phase. Let us first see our first diagram of cell cycle that is shown here. You can see this is the interphase. Interphase is the preparatory phase for the cell division. All the preparations happens during the G1, S and G2 phases. This is M phase that actually divides the cell in two parts. This M phase is further divided into mitosis and cytokinesis. If we talk about the mitosis, it is again divided into four phases, that is prophase, metaphase, anaphase and telophases. Cytokinesis is the step at which step the cell actually divides in two phases. It is of two types, whether it is happening in plants or it is happening in animals. It will depends that how this cell will divide. Here you can see the G0 stage is also shown in this diagram. If the cell escapes from the M cell, that is from the mitosis stage, it comes in high specialization. For example, you can see the nerve cell is shown here. This nerve cell is a highly specialized cell and it is in a G0 stage. That's why most of the time our nerve cells does not divide. So here in G0 phase, no active division is present, but in any requirement, these can come into the interphase and these can divide if they are forced to do so. Let us see our this cell cycle in detail. If we talk about the 6 to 8 hours, it comes our S phase. G2 phase takes 4 to 6 hours. We are taking 24 hours as a normal cell cycle. So in 24 hours there will be 8 to 10 hours for the G1 phase, 6 to 8 for S and 4 to 6 for G2 phase. The remaining 5% time goes to this mitosis or M phase. So let us see one by one what is happening in all these phases. First one is G1 phase. In the G1 phase, the cell is metabolically active. It duplicates organelles and cytosolic components. Also, it starts replicating centrosomes. As we know that centriole duplication occurs in this particular phase. Let me write here. Centriole duplication will occur in G1 phase. So this is all about the G1 phase. Coming to the next, our S phase. S phase is termed as synthesis phase. The name S has come from this synthesis. In the S phase, cells synthesize its DNA. DNA is replicated. In the next phase, that is in G2 phase, that lasts for 4 to 6 hours. In this stage, cell growth continues. Enzymes and other proteins are synthesized and replication of centrosome is completed. Coming to the most crucial step of the cell cycle, that is M or mitotic phase. This mitotic phase is divided in 4 phases: prophase, metaphase, anaphase and telophase. Prophase is a phase in which the nucleus membrane, all organelles, etc. degrades. In the metaphase, the chromosomes align on a central plate like this. You can see these are the chromosomes that are aligned on the central plate, and this central plate is known as a metaphasic plate. If we talk about the anaphase, so anaphase, here you can see these are spindle fibers that start attached to these chromosomes and they start to spread towards the tails. Stretching of these spindle fibers occurs. And if we talk about the last one phase, that is telophase, so in the telophase, something like this, these chromosomes reach to a pole. Here almost 2 cells are present, and in the process of cytokinesis these 2 cells will divide and further the 2 cells from one cell will form. So this is all about that how our cell cycle is happening. Here you can see this is one cell, and after mitosis there are 2 cells that are equal in shape and size. So here in this diagram you can see that some cyclin dependent kinases complex is given, that is cyclin and CDK complexes that are responsible for the regulation of cell cycle. So let us first see that a checkpoint that is present between G1 to S phase. This will regulate the transition of G1 phase to S phase. This is cyclin E cyclin and CDK2. This is the complex that will regulate the transition of G1 to S. If there is any error, then this cycle will halt from here, and this cyclin E and CDK2 will stop the progression of cell cycle. If everything is normal, then cell cycle will proceed to S phase, and this will require the transition from S to G2 phase. A cyclin A and a CDK2 will regulate this transition, that is from S to G2. If there is any error, then again this cycle will stop, but if everything is normal, then this cycle will proceed. The checkpoint that is present between the G2 and M phase is cyclin B and CDK1. These are responsible for the transition of G2 to M phase, and again for the initiation of the cell cycle from transition from M to G1 phase. This will require cyclin D and CDK2. Recently, it has been clear that the cell cycle has several checkpoints mechanism during which the cell stops its progression throughout the cycle while it repairs damaged DNA. Whenever there is any error in the replication of DNA, or any kind of like mutation or any other error, the cell cycle will not proceed. It will halt, and firstly it will repair the DNA, and again it will divide. If there are no possibilities for the repairing of DNA, then cell will ultimately go to apoptosis. Apoptosis is a process of cell death. The activity of the cell cycle varies depending on the status of the cell and the cues such as cytokine stimulation. This cytokine stimulation the cell receives from its environment. Next point, the epiblast cells of the post-implantation E5.5 to E6.0 mouse blastocyst, for example, have a mean cycle time of 11.5 hours. But a day later, at E6.5 to E7.0, epiblast cells have a mean cell cycle time of only 4.4 hours. What factors regulate the cell cycle during development, or how this cell cycle alters gene expression or any other event in embryogenesis? This remains largely unknown. Cell death is a normal process during embryogenesis. Cell death, that is also known as apoptosis. This cell death is of two types, that is, it can be natural or it can be induced. If we talk about the natural cell death, then it is known as apoptosis, and if there is any injury and just because of any injury, if there is cell death, then it will be known as necrosis. So let us discuss these two kinds of cell death. First point, The process of cell death can occur by necrosis or apoptosis. The latter is a form of a genetically controlled cell death, that is, the apoptosis. It is also known as programmed cell death or natural cell death. This apoptosis in itself is an important aspect of normal embryonic development in vivo. This is happening in vivo, that is inside the body. When the genetic program for apoptosis becomes activated, the cell commits a form of molecular suicide. This will be termed as necrosis. Necrosis is not a natural type of cell death, it is induced type of cell death that will be induced by the cell itself. Next point, as the body of an embryo develops, apoptosis helps shape it. Apoptosis has many functions in body development, as if there is a requirement of any gap like between digits or any kind of gap generation and formation of a proper shape, then the apoptosis will help in. If we take an example, so if we talk about the apoptosis, so apoptosis helps control the spacing of nerve cells in the brain and spinal cord. It also helps generate the space in the middle ear, the space that is present in the middle ear. This is generated by the apoptosis, and apart from this, many other organs that are shaped by the apoptosis. Many of the genes that regulate apoptosis were discovered in studies of the microscopic roundworms like C. elegans. The mammalian counterparts of these genes are very similar in terms of their DNA sequences and are called homologous. Next point, in C. elegans, the C84 and C83, or we can say that STAT4 and STAT3, these are two genes that are activated in that order prior to apoptosis. They in turn activate enzymes called caspases. Caspases are the enzymes that are responsible to regulate the process of apoptosis in case of C. elegans. So if we talk about these caspases, these are enzymes which actually triggers apoptosis. These are responsible for the apoptosis to happen. DNA methylation and genomic imprinting affect embryonic development. How it affects, let us see. The very first point is DNA methylation is the process of adding methyl groups to specific cytosine residues. Remember, this is cytosine on which the methyl group will be added. in the promoter regions of DNA. Although DNA methylation is a genome-wide phenomenon. Next point. When the methyl groups are bound at their designated sites in DNA, the transcription factors cannot bind to the DNA and the gene transcription is turned off. Also, DNA methylation causes a rearrangement of the structure of chromatin, the combination of DNA and protein that forms the chromosomes. DNA methylation patterns change during development and their rearrangement in different tissues at different times is an important method for controlling gene expression. Next point. Also important to embryonic development is the process of genomic imprinting. This genomic imprinting causes certain to genes turned on or off. Genomic imprinting is a process that can activate any gene or it can also inhibit any gene or any transcription factor. This is responsible for the on and off or these some transcription factors or genes. The function of these genomic imprintings depending on whether they are inherited from the mother or the father. For most of the genes known to undergo imprinting, specific regulatory regions have been identified where methylation takes place. Methylation does not happen at any place in the DNA. There are fixed and limited sites on which methylation occurs. So this was all about methylation of DNA and the process of genomic imprinting. These two processes have a very large effect in the development of embryo. So thank you very much guys for watching this session. Follow ITLS Academy on various social media platforms and also subscribe our YouTube channel ITLS Academy. Thank you very much everyone for connecting with ITLS Academy and watching this session. Thank you.